this is Roger Green, host of the Surfing the Mesh Tsunami podcast. This week, we're offering 4 conversations from episode 19, our interviews with Mike Patel, Louise Campbell, and Sven Francque about their impressions of the EASL Congress meeting. Plus, instead of a vault, a special excerpt we cut from Louise's interview so that we could get the overall episode down to an hour and 10 minutes. It's got its own content, its own topic, and it's pretty powerful in its own right. This conversation took place with Sven Francque 90 minutes after the final gavel of EASL Congress 2024. It focuses on plans and hopes for the future. We start the conversation by discussing all the positive energy coming from the Congress, not only for Mazel, but for the entire liver community. Sven describes the ambience of the meeting as feeling like a, and I quote, vibrant hepatology community, end of quote, with a lot of interaction. He goes on to discuss the CPG guidelines, a committee on which he was a member, and please note we will cover on episode 21, posting on June 27th. And he also discusses the vision of disease that led to a broad forward-looking guideline update. We go on to cover a set of positive results from earlier stage clinical trials, with Sven commenting specifically on tirzepatide, efruxifermin, and an array of positive phase 2 trials, one of which is lanifibranor, which had presented its results previous year. Sven also found 2 studies that used different methodologies but evaluated the prognostic value of liver stiffness to be important. He discusses the value of the new nomenclature and how it allows researchers to look more broadly at the entire steatotic liver disease category. And we close by talking once more about how positive the body of drug development studies were. As you've heard already, and we'll hear repeatedly over the next month, this was an extraordinary meeting. These conversations should give you a flavor of the broader discussions we will offer in the next several episodes, starting with drug development on June 20th. Hi, so we're here with Sven Francque. Sven, congratulations on an amazing conference. First, I want to congratulate you on a fantastic meeting, and you were part of the 2 big events, which we'll talk about, the CPG guidelines and then the late breaker session. But how did you find the general mood of the meeting? How did it feel in Milan?
Sven Francque.
It really felt like a vibrant hepatology community. I mean, there was a lot of very interesting stuff, both in terms of new science presented, an excellent postgraduate course, Basic Science Symposium. So I think there was really a lot to discover and very interesting content, but also the ambience was really, really nice. It really felt like a vibrant hepatology community with a lot of interaction, networking. Walking around the conference center, you felt the positive ambience, and people were in a way happy to be there and part of the meeting and participate and absorb not only all the content but also interact with each other. Yeah, it felt very well. Yeah, it was really a nice experience.
Well, it felt really engaging and powerful from over here. Even the energy made it all the way from Milano to Pennsylvania, which is great.
Well, then it must be very strong.
It was quite strong. Let's tackle a couple of things one at a time. Okay, first of all, I know you're part of the committee that wrote the CPG guidelines. I believe, and the people I've spoken with believe, that they were fantastic and bold and badly needed. What do you believe was the most compelling part about the guidelines, or how they were put together, or the overall message they deliver?
There are several elements, First of all, I think the important thing is that we based it on the new nomenclature. And to me, the most important thing there is that we clearly state that for those chronic problems, you need to look at all potential risk factors for chronic liver disease. It's metabolic, of course, pointing towards the root cause of this disease topic in the CPG, but you have to have a broad vision and a holistic approach in that sense. Another point is that, again, as the 2016 guideline, it was a collaborative effort with 3 societies. It's another way of pointing out that this needs a holistic approach and that the liver is embedded in, but also a motor of metabolic abnormalities. Another element, I think, is that we were quite more pronounced on what to do in terms of case finding and how to do it. And then also in terms of treatment, we emphasize the need for a multidisciplinary treatment. We tackle what can already be done with currently available drugs, not specifically licensed for NAFLD, MASLD, but still useful in that population within their proper indications. And we already incorporate also the fact that resmetirom is FDA approved, accelerated approval, and we incorporated that already in the guidelines. So it's a little bit future-proof, I would say.
So it makes the guidelines something that people can use in a year or two. I think that's wise. I think that's a very wise thing to have done. Did you get any pushback on any of the items that you just talked about, or were there areas of tremendous debate while the guidelines were coming together that you're comfortable discussing at a high level?
There were for sure areas of debate, yes, but that's, I think, also the added value of doing this together with three societies and have the view of the different specialties that have to take care of people living with this disease. The issue that I mentioned about case finding and how to do it was for sure something that has been debated. The guideline is in line with the other guidelines, but is a little bit more granular, as it does not stick to one threshold, but it uses a rule in and a rule out threshold. You could go for a more simplified version with just one threshold. It's a little bit more granular than that.
So certainly CPG was one of the major important elements of the conference. Another was a lot of work on drugs. And as the guy who co-chaired the late breaker session, I would love for you to comment on, first of all, the two drugs that were mentioned there, the two MASH drugs that were mentioned there, tirzepatide, efruxifermin, and then on some of the others that were in the general session or that you thought had really interesting work presented, or resmetirom for that matter, actually.
If we first take the things that were discussed today at the late breaker, of course, you have tirzepatide, quite impressive results. The MASH resolution result is, of course, very impressive, also clear dose-dependent effect. The results on fibrosis are a little bit puzzling, also looking at the publication that became available. Numerically, you also see clear effects, but there, of course, you have the different ways of presenting it with including patients that have dropped out for several reasons. And it's a little bit puzzling that there is no dose-dependent effect there, although there seems numerically to be an effect. So the small numbers would tend us to not overemphasize, but anyhow, this is encouraging. I mean, having 50% regression of fibrosis compared to placebo with, according to the interpretation, 25 or 30%, still it makes a huge difference. And with all the caution with the low numbers and the way the data are analyzed and presented, still it should also be puzzling if you have such a powerful effect on MASH. It would be puzzling if you don't have an effect on fibrosis in terms of fibrosis regression. So I think this is something that in a phase 3 trial will be well established.
So that's tirzepatide, and then is efruxifermin also, right?
Yeah, efruxifermin. So the 96-week data that Vlad has presented, I think that's also very interesting. I mean, there are several interesting aspects there. First of all, you see that patients that on drug treatment had a response on week 24, they maintained that response, which was not the case with all the placebo patients that had a response. Some of them lost the response on the long term, so the 96 weeks. That's the first important observation. A second important observation to me was that, of course, you added a number of patients that have a treatment response with longer treatment. So longer treatment results in more patients achieving the response in terms of fibrosis regression and also MASH resolution. Although it was also interesting to see that the majority of responders is still already occurring quite early. So in the first 24 weeks, already a lot of responders are responders. And you add more responders with longer treatment, but still more than half of the responders is in the first 24 weeks of treatment, which is also interesting to see in terms of understanding the dynamics of the disease and whether we observe is MASH resolution and subsequent fibrosis regression or some direct fibrosis effect. You cannot really tell from the data, but at least it suggests that there might be some direct anti-fibrotic effects, explaining that still the majority of patients with a response are occurring in the first 24 weeks of treatment.
Interesting you should say that, because Vlad made a comment in response to a question today where the question was about level of fibrosis regression versus level of MASH resolution. And his answer was, well, maybe there's direct fibrotic effect. So that's now you saying the same thing for a second reason. That's fascinating.
Yeah, sure.
So I want to go back to a couple of things. Number one, how exciting it is that we've seen so many drugs come through different modes of action come through phase 2 here.
Yes, absolutely. And also because some of the data are quite convincing. Again, we should be cautious with the interpretation of the data because sometimes it's low numbers. The way people that drop out for any reason during the trial and don't have their end of trial biopsy are handled also has an impact on the precise figures that we see. So some caution is always needed with the interpretation of this kind of phase 2 data. But anyhow, you see impressive results on MASH resolution. You see results that are quite suggestive of a clear effect on fibrosis also. And then we have also the previous data with lanifibranor also showing a strong antifibrotic effect in 24 weeks time. So there are a lot of potential drugs in the pipeline, and we'll see with phase 3, but still this seems to be quite encouraging to have after the phase 3 trials, different compounds available with different modes of action, which then offers avenues to care more patients because there are always non-responders. So there might be treatment options for non-responders to drug A with drug B or combination treatments. It's not for tomorrow, of course, because we have to do all the work with the phase 3 trials. But still, it means that now the field is really moving and that there is at least some potential that within a few years we will have different treatment opportunities to combine or to have alternative treatments if patients are not responding. We're not there yet to have clear indicators of what would be the ideal population for drug A compared to drug B. That is still something we need to work on.
So Sven, it's just my personal experience when you describe exactly what you just did. I first met this disease about 5 years ago at NASH-TAG 2019. And at that point in time, there were 2 drugs that had spotty phase 2s going into phase 3, and we were hopeful. But people were optimistic, but a lot of that was wish and hope. And the question was, how do we get fibrosis down one level in a clinical trial? We're now 5 years later. We have a drug on the market. We have some clearly affirmative phase 2s that have answered some interesting questions. One of the interesting things to me about the efruxifermin 96-week data was that pegbelfermin was promising initially and then fell back over time, which was why it failed. This obviously is going to happen here. So you look 5 years forward and you can see several approved drugs, I think, I hope.
That's a realistic thing, yeah. That's a realistic thing. I think some of the results of the earlier phase 2 trials, and that's always the caveat with phase 2 trials, large numbers and the way data are presented. Some of the phase 2 trials that were positive, they were positive, of course, but the results were not always as compelling as the results we see now. But that's also the way it progresses and what we have seen with other diseases. In the beginning, you see small effect sizes and you are happy, and then you get better and better drugs with larger and larger effect sizes. I mean, that's how it goes.
That's what comes next. Also, there was a lot of mention in CPG about ways to use NITs to classify patients, and some NIT work in the meeting itself. Is there anything you'd like to comment on that front?
The interesting thing, I think, is that there are 2 studies about, they're not completely the same, but 2 studies about the prognostic value of liver stiffness. And I think that is one of the important ways to go, and one of the gaps that we have today is how to link changes in NITs over time with the prognosis of the patient. And in particular, that study with Vincent Wong, I think, as last author, about changes in liver stiffness, in this case with VCTE, 30% up or down, that evolution is quite predictive of prognosis of the patient. And I think that is, to me, one of the main progress we are starting to make in terms of the interpretation and working with NITs. It's a better understanding of what their evolution over time can tell us. And I don't think we're completely there yet, because it's not always as accurate on an individual patient base, but still, I think it's an important piece of the puzzle that we were missing and that we are step by step discovering.
Okay, so is there anything else about the content of the meeting that we've not covered yet that you'd like to speak about for a moment or two?
MASLD, I think these are really the most important issues. Another interesting observation was also, and it's a little bit broader than MASLD, it's SLD. It was a presentation by Mats Israelsen from Alexander Krag's group about how over time patients people can switch from one category to another, and that some of the MASLD patients after a reevaluation weren't MASLD anymore or were MET-ALD and vice versa, showing how dynamic that classification potentially is. There are two caveats there. First of all, the diagnosis of steatosis was based on CAP, and we know that CAP, especially in terms of precisely quantifying steatosis, is probably not always as accurate. It's especially not in follow-up. So it needs also, I think, another modality of diagnosing steatosis to confirm these data. And then, of course, alcohol was self-reported, so that's also a caveat. But nevertheless, it shows you how dynamic it is and how important it is to really take into account not just cross-line diagnostically, but also in follow-up of patient, how different factors that cause steatotic liver disease, whether it's alcohol or cardiometabolic risk factors, can evolve over time, and those evolutions over time also impact on the liver. And so the history of that will also be important if we assess a patient. But otherwise, I think we covered not everything, but the most important highlights. We did not specifically talk about survodutide, but also there you have results that are a little bit comparable with tirzepatide.
Or maybe even a little bit stronger, we'll see. But yes, and I think it was exciting that both different modalities of double incretin agonists both showed successful results. I think people were always confident about GLP-glucagon, less so about GLP-GIP. So I thought tirzepatide in that regard was good news, really good news.
Yeah, indeed. But with the caveats that I mentioned. Yeah.
So, Sven, thanks for joining us today, and been through a lot. I'm sure you're pretty tired by now, so thanks again for joining today.
And now, back to Roger.
We hope you've enjoyed this recording. If you have any questions or comments about the content of this conversation or the entire episode, please put them in the review section of the page from which you downloaded this conversation, or send an email to questions@surfingmash.com. Until then, stay safe, surf on. We'll see you on the podcast. Bye-bye now.